Okay, I think we're going to go ahead and get started. So thank you for coming to another session of Nano at Tech. Before I introduce today's speaker, just a word about the next session, which will be actually 3 weeks from today on March 29th. We're avoiding spring break here at Georgia Tech. So on March 29th, we'll be back in the Marcus building, and our speaker will be Professor Leslie Chan from Biomedical Engineering here at Georgia Tech. So it's a real pleasure to have with us today a visitor from off campus, Professor Jin Xie from UGA. Jin got his bachelor's degree at Nanjing University and then his PhD in chemistry at Brown before doing a postdoc at Stanford and then following his postdoctoral advisor to the NIH to finish up his postdoc. And then went to UGA starting in 2011, where he's currently an associate professor of chemistry with an adjunct position in chemical materials and biomedical engineering. He's won a number of awards, including an NSF Career Award and NIH Pathways to Independence Award. And he's also an associate editor of the Journal of Nanobiotechnology. And with that, I will turn it over to you.
Thanks for the kind introduction. Great pleasure to come here and speak to you about some of our recent research progress. The title of my talk today is Alkali Halide Nanoparticles as a Cancer Therapeutics. Disclosure. So this is my plan for today's talk. I will start by telling you how sodium chloride nanoparticles can be used as potential cancer therapeutic. And then I would like to switch the gear a little bit and talk about the different technology called X-ray induced photodynamic therapy, or X-PDT, and how another alkaline halide nanoparticles, cesium iodide, can help X-PDT. And lastly, I would like to mention yet another alkaline halide nanoparticle, potassium iodide nanoparticles, and why they can be used as a radiosensitizer. So if you have been paying attention to progress in nanobiotechnology field, you may notice how many elements have been made into nanoscale particles. We have conventional carbon nanotubes, silicon nanoparticles, calcium nanoparticles. We have magnetic nanoparticles made from transition metals. We have noble metal nanoparticles, quantum made of boron, oxygen, and nitrogen group elements, and we have commercial nanoparticles made of lanthanides. But very few have looked at alkaline and halides nanoparticles. Why is that? Well, there are probably two reasons. One is that this alkaline halides compounds are highly water soluble, so that makes it a little bit more challenging to make these nanoparticles. And second, and what's the point, right? So taking sodium chloride, for instance, and even if you can make these nanoparticles, once they get into water, they're going to degrade and become salt and just function as a salt. So what's the point of making these nanoparticles in the first place? Well, if you really think about it, for all mammalian cells and across the plasma membrane, you have a sodium gradient. You always have a relatively low sodium level relative to the extracellular sodium level. Potassium is the opposite. And this ion gradients are essential for cell life and functions. Actually, cells spend up to 1/3 of its energy maintaining that gradient. In general, osmotic balance is critical to again cell functions and volume and life. If you take a cell and put them in a isotonic solution like water, cells gonna swell and burst, right? And because you break the osmotic balance, you reduce the extracellular osmolarity. In theory, if you can do the opposite, you can increase the intracellular osmolarity. You can also break the osmotic balance to increase the cell volume and probably cause cell lysis. But that is much more challenging because the plasma membrane is not permeable to ions. if you simply apply sodium chloride salt, you will increase the extracellular sodium level, but minimally affect the intracellular sodium level. So that leads to our hypothesis. If you can make sodium chloride nanoparticles and somehow protect the particles from fast water degradation, so these nanoparticles can get into cells through probably endocytosis and then degrade inside of a cell, and they will release large quantities of sodium and chloride, and that potentially will break the osmotic balance and kill cancer cells. So that's our goal here. That's our hypothesis. So how to prepare sodium chloride nanoparticles? So obviously, you don't want to do that in water. We actually tried different recipes, and we end up using solvents like a mixed solvent of hexane and ethanol. And we use, for instance, sodium oleate and acetyl chloride as the sodium and chloride chlorine precursors. And we typically use some surfactants like oleamine, which can passivate on particle surface. So you have an alkyl chain coated on nanoparticle surface, coated on nanoparticle surface. You can play around with the reaction conditions. You can make particles all the way from 15 to 500 or even larger sizes. And we did XRD and EDAX, and we confirmed that it's sodium chloride that we got. So again, the as-synthesized nanoparticles are coated with a layer of alkyl chains, and the surface is hydrophobic. So we did surface modification using amphipathic compounds, in this case, a pegylated phospholipid. The phospholipid has these two alkyl chains and bind to particle surface through hydrophobic interaction. And you have a PEG chain along with the amine head group stick outward, and that helps particles to be suspended in water. So after surface modification, the particles are temporarily stable in water. The surface is a little bit positively charged, which helps particles to get inside of the cells. And we also looked at the nanoparticle degradation in water. So the coating slows down, but does not prevent particles from water degradation. The half-life is about 5 to 6 hours, which we think it's reasonable for our application, because you don't want the particles to be too unstable. That means particles are going to degrade before entering cells, and then they won't be toxic. And you don't want the particles to be too stable either, because that means particles will get inside but won't release sodium and chloride. So we think this particular formulation is fine for our applications. So then we started testing these nanoparticles in vitro. We first labeled the nanoparticle with a dye molecule, and we looked at their cellular uptake. And we also did the lysosome tracker, so co-stain endosome and lysosome. And we found there's a pretty good overlap between the nanoparticle and the lysosome tracker. Basically means that particles are getting internalized through endocytosis. And we also looked at the intracellular sodium level change. We used a dye called SBFI-AM that is responsive to sodium. And we found that in cells treated with our nanoparticles, there is a time-dependent increase of sodium level. And if you treat cells with sodium chloride salt at the same concentration, we don't see significant sodium level change. We also looked at the nanoparticle uptake and sodium level change, and we found a pretty good correlation. And basically, this studies confirm our hypothesis, which is that particles can get internalized through endocytosis and break down and release sodium, and that increase the intracellular sodium level. We actually looked at the chloride level as well, and we found it was also increased. And then we tested particles. We looked at the cytotoxicity of these nanoparticles, and so we were here. So we then looked at the cytotoxicity of these nanoparticles. We started from PC3 cell, which is the prostate cancer cell line, for no particular reason. We just happened to have the cells in culture. And we found that these nanoparticles are indeed toxic, and the IC50 value is about 160 microgram per milliliter. So as a comparison, medical saline, 0.9% medical saline has a sodium chloride concentration of 9,000 microgram per milliliter. And we then looked at the impact of this nanoparticle treatment on intracellular and organelles and components. And we found that in cells treated with nanoparticles, there is clear damage to mitochondria. We see a depolarized mitochondria, and we also see reduced metabolism rate and decreased the production of ATP. And that in turn leads to increased intracellular reactive oxygen species level. And that elevated oxidative stress was also confirmed by Western blotting, looking at some typical oxidative stress markers like ERK and P38, and which are all activated. And that leads to extensive damage to cell DNA and lipid peroxidation. We see an increased level of double-strand DNA breaks and elevated the lipid peroxidation. And that culminate to induce cell death. We see a cytochrome C release, which is a trigger of intrinsic cell apoptosis pathway, and that and the apoptosis was also confirmed by elevated caspase-3 activity. So basically, nanoparticles able to induce cellular apoptosis and kill cancer cells. And we also did imaging study, dynamic imaging study, looking at the cellular volume change and at the early hours of nanoparticle incubation. And we found that at relatively high nanoparticle concentration, so shortly after the incubation, within 6 hours, we see a significant increase of cell volume up to 50%. And we also find that some cells, are going through lysis and over the incubation. And so basically cells die of necrosis at early hours at relatively high nanoparticle concentration. That necrosis was also confirmed by PI staining and LDH study. So basically nanoparticles are toxic. They can induce cell apoptosis, and relatively high concentration cells also die of necrosis. And that is PC3 cells. We also tested the particles with other cell lines, including breast cancer, bladder cancer, and lung cancer cell lines. And we see IC50 values range from 50 to about 200 microgram per milliliter. And again, sodium chloride salt at the similar concentrations is not toxic to cells. And also, if we pre-incubate nanoparticles before we add in particles to the cell culture media, and we see reduced cytotoxicity. So again, particles need to get into cells to kill cancer cells. If they degrade and become salt, they lose their potency. If you pre-incubate nanoparticles for 8 hours or longer, they lost the majority of their toxicity. We also tested the nanoparticles with normal cells, and including like primary urothelial cells and the prostate epithelial cells or stem cells, and we found that these cells are much more resistant or much less susceptible to sodium chloride nanoparticles. And there are only 2 reasons to the selective toxicity to cancer cells. One is that cancer cells have a relatively high endocytosis rate, so they tend to take up more nanoparticles. That was confirmed by flow study, and we compare nanoparticle uptake between normal cells and cancerous cells, and we found that cancerous cells take up more of these nanoparticles. And the second reason is that cancer cells have a higher sodium baseline to start with. This was actually well documented back in 80s and 90s. A lot of groups study the intracellular sodium level, and they found that faster proliferating cancer cells has the baseline sodium level that is about 2 or 3 folds higher than normal cells. We looked at the cell, again, baseline sodium levels and their IC50 values, and indeed we found that there's a good correlation between the baseline sodium level, high baseline sodium level, and the susceptibility of the cells to sodium chloride nanoparticles. So both factors contribute to the, selective toxicity to cancer cells. Then we tested this nanoparticles in vivo, started with some subcutaneous models and established on immune compromised animals. And for control, we injected the saline at the same sodium chloride level. And we found that nanoparticles can indeed slow down tumor growth, inhibit tumor growth, and cause extensive cell apoptosis after the treatment. And we did not find any toxicity, which is not surprising. This is actually one of the benefits of using sodium chloride nanoparticles as a treatment. After killing the cancer cells, they're going to degrade and become salt, and that is going to be either absorbed by the host or safely excreted. So we don't have a long-term toxicity issues here. Yet another interesting property of sodium chloride nanoparticles is that they can cause this so-called immunogenic cell death. So traditionally, we think that most chemotherapies cause cell apoptosis, which is considered a tolerogenic or non-immunogenic pathway. The cells that die then become apoptotic bodies, and that is absorbed, that is taken by neighboring cells or macrophages, and they won't trigger immune response. Recent studies show that some chemotherapies and radiation therapy or photodynamic therapy can cause this so-called immunogenic cell death, meaning that not only kill cancer cells, but also release tumor antigens and then promote the secretion of some dangerous signals that can, for instance, recruit dendritic cells and activate dendritic cells and promote their antigen presentation to T cells. In turn, leads to a boosted immune response against tumors. So basically, you not only kill cancer cells, but also help stimulate the systemic anti-tumor immune response. So our sodium chloride nanoparticles, because they can cause necrosis, which is a highly inflammatory process, our hypothesis is that they can also induce immunogenic cell death. And we looked at some typical ICD markers, including ATP, HMGB1, and CRT. And we tested particles with different cell lines, and indeed we found that that the cancer cells killed by sodium chloride nanoparticles, we see elevated secretion of ATP and HMGB1, and increase the surface presentation of CRT. So again, confirming that these nanoparticles can cause immunogenic cell death. We also tested this in vivo. We did a vaccination study, basically. We kill cancer cells with sodium chloride nanoparticles, and then we inoculate these dead cells to vaccinate animals with this dead cancer cells. And then we inject live cancer cells to the contralateral side of the animal. And we monitor that tumor establishment and growth. And for control, we vaccinate animals with cancer cells that are killed by freeze and thaw, several cycles in freeze and thaw. And we found that animals that are vaccinated with cancer cells killed by sodium chloride nanoparticles, indeed are more efficient at rejecting this live cancer cells and or slow down their tumor growth. Again, we attribute this to the ability of nanoparticles to induce immunogenic cell death. So that is all nice, but what about the real world and application? So currently we're focusing on non-muscle invasive bladder cancer. The bladder cancer is the sixth most prevalent cancer type in the world, and about 75% of bladder cancer patients are diagnosed with early stage non-muscle-invasive bladder cancer. The typical treatment is transurethral resection to remove the majority of the tumor, but the recurrence rate is very high, up to 70%. So the surgery is usually followed by so-called intravesical therapy with either chemotherapy or BCG, which is a vaccine. And BCG is the preferred treatment. But the problem is that about 30% of the patients do not respond to BCG. And chemotherapy as the second-line treatment is not effective. So our hypothesis is that our sodium chloride nanoparticles can be used as an intravesical treatment. So we'll deliver nanoparticles through urethra and directly applied to the affected bladder. So the particles can kill cancer cells, and after that, they're going to degrade and become salt, and that's going to be excreted by urine. Another hypothesis that we had is that we can probably combine this local nanoparticle treatment with immune checkpoint inhibitors, so immunotherapy. So you probably heard that there has been a lot of progress in immunotherapy, this antibody-based immune checkpoint inhibitors that are now used in the clinic for management of different types of cancer, including bladder cancer. But not all patients respond to immunotherapy. Some patients have the so-called cold tumors. So our hypothesis is that by combining our nanoparticles, which again is a highly pro-inflammatory treatment, we can combine the local nanoparticle treatment with systemic immune checkpoint blockade to achieve more sustained and tumor control. So we started from some subcutaneous tumor models. These are syngeneic tumor models established on immune-competent animals. And we found that indeed our sodium chloride nanoparticles can effectively inhibit tumor growth, and the treatment is more effective when the particles are used in combination with anti-PD-1 and antibody treatment. And we also looked at infiltrating immune cells into tumors, and we see elevated we see that in tumors treated with nanoparticles or nanoparticle antibody combination, we see increased populations of the mature dendritic cells, cytotoxic T cells, but reduce the regulatory T cells. So in the end, we see an elevated cytotoxic T cell and T regular cell ratio, which are indications that the treatment indeed boosts immune response. So we are happy with the results. And next, we're going to test these nanoparticles in orthotopic tumor models. And on that, we're collaborating with the UNC and Dr. Zibo Li and Dr. William Kim's group. They have this orthotopic tumor model. Basically, you inoculate the tumor to the inner vesicle wall of the bladder of the mouse. And then for treatment, we are going to use a catheter to develop the nanoparticles, directly apply them to the vesicle, to the bladder. So we are testing these nanoparticles right now, and hopefully we can get some good results out of this. So next, I would like to switch the gear a little bit and talk about X-ray induced photodynamic therapy, or X-PDT. And before that, I would like to briefly mention photodynamic therapy, or PDT. PDT is an interesting and relatively new cancer treatment modality. The essential component including photosensitizer, light, and oxygen. So photosensitizer would be a molecule like a porphyrin or phyllocyanins, and those compounds are not toxic in dark, but when they activated by light, they can be activated to a triplet state, and from there they can activate a nearby oxygen molecule and produce radicals like a singulated oxygen, which is toxic. So it takes the combination of photosensitizer and the light to produce toxicity. PDT is effective and it's also highly selective because you can choose where to give light. The main limitation of PDT is that visible light has a limited tissue penetration ability. So even in a near infrared spectrum window, we are talking about effective penetration depths of less than 1 centimeter. So that has been a major restriction of PDT and the limits its applications in the clinic. So we and others are trying to address that issue and by developing a technology called X-ray induced photodynamic therapy. Some call it the radiodynamic therapy or RDT, that is pretty much the same technology. So the idea is to use X-rays, which afford much greater tissue penetration ability and widely implemented in the clinic to activate a photosensitizer. And for that to happen, we need a transducer. So we're using scintillated nanoparticles that can down-convert X-ray photons to visible photons, which can in turn activate a photosensitizer. So typically, you have a complex nanoparticle that encapsulate both the scintillator and the photosensitizer. The photosensitizer should have an excitation that matches the emission of the scintillator. So that's, again, the idea is that if you can deliver that to cancer cells, and then on the X-ray, the scintillator is going to generate X-ray luminescence, which in turn activate the photosensitizer, produce toxicity. So we started testing this idea back in 2015. We started from a scintillator. It's a European doped strontium aluminate. My collaborator, Dr. Zhengwei Pan at the UGA, made this nano ribbon, strontium aluminate nano ribbon, but it's too big for our applications. So we have to go through mechanical grinding and sediment to get particle to bring the material size to the nanoscale. This top-down approach is not the best at the controlling particle size. But anyhow, we get particles from 100 to 200 nanometer, and we confirm that these particles can produce luminescence under radiation, and it's centered about 510, 520 nanometer. And then we coated these nanoparticles with a layer of mesopore silica. So basically, the silica layer is porous and has the channels throughout the silica layer, which can physically trap or encapsulate small molecules. So we loaded into the mesopore silica layer a photosensitizer, MC540, whose excitation matched the emission of strontium aluminate. And then we tested these nanoparticles first in solution. We apply X-ray, and we found that singulated oxygen is indeed produced. So basically confirms that X-PDT is activated. We also tested these nanoparticles in vitro, again in cells, for cells that are treated with these nanoparticles under radiation. We see elevated level of singulated oxygen, and that leads to cancer cell death. And we also tested these nanoparticles in vivo. So for proof of concept study, we directly injected the particles into tumors, and then under X-ray, that treatment was able to pretty effectively arrest the tumor growth and even cause tumor regression in some cases. We also tested other scintillated nanoparticles. This is in 2017, and we used this the chromium of the lithium halides that was synthesized the first by social chemistry, and then we have to use bone milling to again bring the material size down to the nanoscale. And then we coated the particles with again mesopore silica, and we loaded a different photosensitizer, NC, that matches the luminescence of the particles. One thing that is interesting about this lithium halides is that its emission is in the near-infrared region. So that means, relatively speaking, that luminescence has a better tissue penetration ability. So we can keep track of these nanoparticles inside of a small animal by just monitoring that X-ray luminescence. And this time we tested the particles in an orthotopic lung cancer model. We conjugate onto the nanoparticle surface an anti-EGFR antibody. EGFR is a protein that is upregulated in different in many types of cancers, including lung cancer. And we found that for particles that coupled with the antibody, the nanoparticles are able to selectively accumulate in tumors in the lung. And then if the particles are not conjugated with antibody, we see a much less uptake in tumors. And then on the radiation, external radiation applied to the lung area, 6th grade, we see a pretty effective the treatment, improved tumor suppression relative to radiation alone. So that's nice, but we see some problems with the current X-PDT nano platforms. So first of all, you need a complex nanoparticle. Then and that is difficult to control, batch to batch quality, and it's difficult to scale up these nanoparticles. Not to mention that our scintillator particles are made from top-down approaches, which difficult to control their size. And another concern is that these scintillated nanoparticles, they contain transition metals or lanthanide metals. So not only us, some other groups have tested the different scintillated nanoparticles, and almost always these materials contain transition and lanthanide metals, which potentially pose a barrier if you talk about the clinical translation of these technologies. There are other issues, for instance, suboptimal delivery of photosensitizer. So this photosensitizer are bound to a large nanoparticle, which is not the best carrier when it comes to delivery large quantities of photosensitizer into tumors. Recently, we switched to a different approach. We are now testing X-PDT with a sodium-doped cesium iodide nanoparticles, another alkaline halide nanoparticle, and 5-AOA. So 5-AOA is the FDA-approved compound. It's an interesting compound, amino acid, and that can be taken either orally or can be injected IV. And it selectively forms protoporphyrin 9 in cancer cells mitochondria. Protoporphyrin 9 is a photosensitizer. It's also a fluorescent compound. So 5-AOA has been used in the clinic for photodynamic therapy, and it has also been used as a fluorescent compound to guide medical surgery and to remove a tumor. And then, so on the other hand, we have the sodium-doped cesium iodide, which is established scintillator, and it gives a strong X-ray luminescence at about 410 nanometer. And you can see that luminescence very well matched the solid band of protein, protoporphyrin 9. So our hypothesis is that this combination can lead to X-PDT. But what's the difference, between this approach and the previous approaches, aside from that you're using a different pair of scintillator and photosensitizer? Well, the difference here is that now we're going to inject these two agents separately. We take advantage of the fact that 5-AOA, again, is an FDA-approved agent, and they can accumulate in tumors on its own. So we just focus on the development of these nanoparticles, and we hope that this streamlined approach will, make it more feasible to scale up nanoparticles, and then hopefully, we can move it forward for clinical translation. Another benefit, of course, is that this sodium-doped cesium iodide, it does not contain the transition or lanthanide metals. they're made of alkali and halides. So after the treatment, they can be degraded and then hopefully can be safely excreted from the host. So this is basically the hypothesis. We're going to synthesize sodium-doped cesium-iodide nanoparticle. We probably need some kind of shell to prevent them from faster degradation. We also need a targeting ligand for the nanoparticles to target the cancer cells. And in this first study, we used the folic acid, and the cognate receptor, folate receptor, is upregulated in different types of cancer. So the particles are going to get internalized through endocytosis, and then on the X-ray, it will generate X-ray luminescence that can activate protoporphyrin that is induced by 5-AOA in cancer cells mitochondria. One thing I would like to add is that we have been talking about X-PDT as if we can convert all X-ray photons to visible photons to activate a photosensitizer. That is not the case. Always have a radiation therapy component, and that mainly targets DNA. So the intracellular distribution of photosensitizer matters. And so previously, photosensitizer is bound to a large particle, and which accumulated on cell membrane or endosome or lysosome, which are known to be less susceptible to photodynamic therapy. But here, the photosensitizer is accumulated in mitochondria, which is known to be 5 or 10 times more susceptible to photodynamic therapy. So we hope that will bring better synergy with radiation therapy and achieve better tumor control. So we were able to make sodium-doped cesium-iodide nanoparticles. It's a nanocube, about 50 nanometer in diameter. So this time we were able to make particles through wet chemistry, so we can control their size and quality well. And we confirm that it's indeed cesium-iodide crystals. And then again, we need to prevent particles from faster degradation. So we coat particles with a layer of magnesium oxide, which is also low toxic and it's not water soluble. And we confirm with PpIX elemental mapping that the coating is on. And after that, we added, again, pegylated phospholipid to convert nanoparticles water soluble. And then this time we have a folate, so folic acid terminal group that is used as a cancer targeting ligand. And we confirm that after the coating, the particle are stable up to 24 hours. So again, we measured the luminescence of the nanoparticles. We found that the magnesium oxide coating does not significantly shift the luminescence of the particle, and which is this red curve here. And then it matches well the solid band of protoporphyrin 9. And then we tested this first in solutions. We mix protoporphyrin 9 and the nanoparticles, and then we apply radiation and we measure the production of singlet oxygen. And we found that significant increase of the level of singlet oxygen in solutions. We also tested the same solution, but put it on a 3 centimeter pork. And we found that the radiation is able to activate X-PDT through that pork block, and that is due to the good tissue penetration ability of X-ray. As a comparison, LED light fails to activate the photosensitizer in this scenario. And then we tested these nanoparticles in vitro, first with 41 cells, which is folate receptor positive. And we found that the particles indeed are able to get into cancer cells if they're coupled with a folic acid particle surface. And then we tested the impact of the X-PDT on cell viability and functions. And for in vitro studies, we use 5-AOA instead of a protoporphyrin 9. And we found that the combination of 5-AOA and the nanoparticles, again, under radiation, were able to significantly elevate intracellular singlet oxygen level. And we actually also see a certain degree of hydroxyl radical level increase. And we also looked at the superoxide dismutase, and we found their activities also increased, indicating that there's oxidative stress in cells after the treatment. And with that oxidative stress, and we see increased level of double-strand DNA breaks and lipid peroxidation. We also see a decreased mitochondrial membrane potential, and we attribute that to the X-PDT damage to mitochondria, and that leads to cell apoptosis, and that leads to reduced cell viability. Again, we first tested this in 41 cells, but later on we tested the same technology in other cell lines, and we see a similar level of viability decrease. And we also tested the technology with clonogenic assay, which is commonly used to measure the capacity of, the potency of radiosensitizers. And we found that 5-AOA and nanoparticle combination at doses while 5-AOA and nanoparticles are not toxic in the absence of radiation, their combination were able to reduce the number of colonies and relative to radiation alone. Then we tested these nanoparticles in vivo, again with the 41 tumor models. We inject nanoparticles to tumors. 5-AOA was given systemically, and we applied radiation 3 gray to tumors, and we did 3 treatments. And we found that the combination was effective at inhibiting tumor growth relative to radiation therapy alone. And we also see improvement in animal survival as well. Actually, after 21 days, after 3 weeks, all animals in the treatment groups remain alive. Meanwhile, we did not see signs of toxicity from the treatment. Actually, in a separate study, we inject nanoparticles IV into animals, and we take the blood and analyze. We did the complete blood counts, and we also looked at the biochem markers like liver and kidney function markers. And so, and we did not see signs of toxicity. And after 2 weeks, we see that the iodine and its DCM have been have fallen to the baseline or close to baseline level. So the next step is to test probably giving the nanoparticles intravenously, and probably also tested the technology in a more clinically relevant models. So lastly, I would like to quickly go through yet another alkaline halide nanoparticles, potassium iodide nanoparticles, and why they can be used as a radiosensitizer. A radiosensitizer is an agent that can enhance the efficacy of radiation therapy under the same physical radiation dose. And for potassium iodide, the key the player here is iodine. We know that iodine molecules have been used as a CT contrast agent, and it's also known that iodine can enhance the photoelectrons that are produced under radiation, and probably more effective on the low or medium energy X-rays. And there actually have been clinical trials testing CT contrast agents and see whether they can enhance the efficacy of radiation therapy. The problem is that the CT contrast agents, the small molecules, tend to clear rapidly cleared from the host and they don't retain in tumor as well. So our hypothesis here is that we can probably use a potassium iodide nanoparticles, which can slowly release iodide. And unlike large nanoparticles, this iodide can efficiently diffuse through the tumor mass. And we also hypothesis that this iodide can be taken up by cancer cells through this sodium iodide transporter, or NIS. So NIS is a transporter that taking iodide along with 2 sodium into cells. It is upregulated in thyroid tissues, and which is why iodide is accumulating in thyroid. And it's found that NIS is also upregulated in some types of cancer, including breast cancer. So basically, we're trying to use the potassium iodide nanoparticles as iodide depot, which can control release iodide, and we exploited the NIS to deliver iodide into cancer cells and to promote to enhance radiation therapy. We were able to make the particles, and then we coated them with a layer of polymer, and that will help control release of iodide. And we test them in vitro. So we see the particles actually don't get into cancer cells efficiently. They're pretty big. But we see, in cells that are treated with the particles, we see an increased level of iodine. And that is because the iodide is going to pump into the cells through NIS. And if you use TRA, which promotes the NIS expression, we see a further increased level of iodine accumulation in cells. And if you use a perchlorate, which is an inhibitor of NIS, we see reduced uptake. So basically, it confirms that iodide can be taken up through the NIS. And we also see elevated cell damage under radiation after the treatment. And we also tested the particle and the radiation combination in vivo, and we see a pretty good tumor suppression. So I give you 3 examples where alkaline halide nanoparticles can be used as cancer therapeutics, either directly kill cancer cells or mediate X-PDT or enhance radiation therapy. With that, I would like to thank you for your time, and my group members and the collaborators and the funding agencies. I'm happy to take any questions from the audience. Thank you.
Questions?
Yes.
So I know for the sodium chloride nanoparticles for the bladder cancer treatment, you inserted them via catheter of the urethra. Is there a way, once they're in the bladder, to control where they go precisely?
Not that I know of. So again, the bladder is a vesicle, muscle vesicle. So basically, it's gonna fuel up the bladder, and then we rely on selective toxicity against the cancer cells to kill them. So, and it's also known that the cells lining the inner wall of a bladder, they have a low level of endocytosis. It's understandable because urine can be toxic. Right? They tend to take up less nanoparticles, so that also helps with the selectivity.
So I guess that leads up to my next question: is there limited depth of these nanoparticles?
That's a good question. I think there have been studies and looking at the size of nanoparticles and their penetration. So in our case, we're using 100 nanometer nanoparticles, but we are able to reduce the nanoparticle size if needed. But that's a good question.
Well, I have a question regarding the X-PDT. Why was the mechanism decided? Why do you need the X-ray to make the energy that's make it effective, and not using the UV or some other light source?
But again, the main limitation is the tissue penetration, right? So If it is a visible light and the penetration effective penetration depth is less than 1 centimeter, so you can treat some probably skin cancer or tumors that are close just under the inner lining, but you cannot use the technology to treat large tumors or tumors that deep-seated. X-ray, on the other hand, has much better tissue penetration ability. But for that to happen, we need a transducer. That's why we use.
So what's the action mechanism? Energy striking, or how do you make it there?
Scintillator is, in general, that's compound that can down-convert X-ray photons to visible photons. So you have X-ray comes in and you generate the visible photons, and that in turn activates photosynthesis.
I actually had a follow-up on that. So for that, for the example you gave, where you inject both the scintillator separately from the photosensitizer, I mean, isn't there a distance dependent to that energy transfer, and how do you get them close enough?
Yeah, that's a great question. So, I think there's a trade-off, and again, if you put them together, there's some limitations. But as you mentioned, if you inject them separately, there's certainly penetration depths and problem. I guess that was our concern, but by testing this in vitro, in vivo, we see that it works. One thing I would like to add, which is that so previously for conventional photodynamic therapy in 5-AOA, people were using 630 nanometer light to activate. So you can see that that band has a low absorbance, and that compromise is needed because lights of photons of short wavelengths is going to be absorbed, or won't have penetrated deep into tissues. In our case, we are targeting the solid band. Its absorbance is more than 10 times greater than the Q band. I think our argument is that you're delivering the light source inside of cancer cells, so eliminate from within, so that that helps mitigate the light attenuation problem. But in the end, so yes, that is definitely a legitimate concern to whether you can activate the photosensitizer now that separately injected. But we see some pretty good.
Certainly the experimental results are positive. Right? Any other questions? All right, it's actually about 1 o'clock, so let's thank our speaker one more time, and we'll see you in 3 weeks.
Thank you.
This.